Hello, my name is Cynthia Rohde, and I'm an Associate Research Fellow at Pfizer. And today I'm going to be talking to you about non-clinical COVID-19 vaccine development during a pandemic. I'm going to start off with a brief background on coronaviruses, and then touch on the target antigen for most COVID-19 vaccines that are in development today. I'll then move on to discuss general safety considerations for infectious disease vaccines in general, COVID-19 vaccines, and mRNA vaccines. Then I'll share with you a brief review of the traditional route of infectious vaccines development and contrast that with the new paradigm for development that we have pursued underneath the COVID-19 pandemic. And finally, I'll close with some lessons learned. Coronaviruses are a large family of RNA-based viruses that infect birds and mammals. They're enveloped single-strand viruses with one of the largest nucleocapsids among the RNA viruses. The name coronavirus, or corona specifically, meaning crown or halo, comes from the crown-like spikes that cover the surface of the coronavirus. These viruses primarily cause upper respiratory and gastrointestinal disease, and this disease can occur in many species, including feline infectious peritonitis, or FIP, in cats, and transmissible gastroenteritis in pigs. The coronavirus, which you're probably familiar with the structure, the envelope consists of 4 major structural proteins, and the little red spikes that you can see on the surface are the spike glycoprotein that mediates viral entry into the cell. In humans, there are 4 coronaviruses that are associated with the common cold, 2 alpha and 2 beta, and more recently, 3 beta coronaviruses have emerged that can potentially cause severe respiratory syndromes in humans. These include SARS, severe acute respiratory syndrome coronavirus, which emerged in 2002, Middle Eastern respiratory syndrome coronavirus, which emerged in 2012, and the virus that we're dealing with today, SARS-CoV-2, which emerged in China in 2019. Among the coronaviruses, the spike protein, glycoprotein, binds to a variety of receptors. In particular, for the human SARS-CoV coronavirus, the original, as well as SARS-CoV-2, the spike protein of these viruses bind to the angiotensin converting enzyme 2, or ACE2. This slide shows an image of the spike protein in more detail. It is a trimeric protein that consists of 3 main subunits. Excuse me, I'm going to go back. And with the small receptor binding domain, or RBD, pointing down and binding to the blue ACE2 receptor on the host cell surface. It is only one of these receptor binding domains. They fluctuate in the up and down position, and when in the prefusion conformation, the spike protein only has one of these 3 receptor binding domains that is in the up position. Now I'm going to transition to telling you a little bit more about general considerations for safety evaluation of infectious disease vaccines. The toxicology assessment of these types of vaccines is intended to evaluate the safety of both the vaccine components as well as the associated immune response. Off-target or severe expected toxicity is generally not accepted, as these vaccines may be administered to millions or even billions of healthy people, although exceptions may be made based on the risk-benefit profile. There are a number of major regulatory guidelines for infectious disease vaccines, and I've listed a few of them here. But there may also be regional and disease-specific guidances to consult. In general, as infectious disease vaccines are most likely targeting a foreign antigen that should not be cross-reactive with any antigens present in humans, non-clinical safety studies that are performed for these types of vaccines are generally smaller in number than for a typical small molecule or biologic. In general, a repeat dose toxicity in a single species is sufficient. And then depending on the target population, a developmental and reproductive toxicity or DART study may also be conducted in a single species. Finally, depending on the specific vaccine modality and if you're using any novel adjuvants or excipients in the vaccine, other non-clinical safety-related studies may be conducted, such as biodistribution, neurovirulence, etc. Some viral infections, as well as vaccinations, and this includes in regards to viral infections, things like flaviviruses, measles, or even respiratory syncytial virus, can elicit neutralizing as well as non-neutralizing, enhancing, or non-enhancing antibodies. If more non-neutralizing or sub-neutralizing antibodies are generated that do not clear or block the infection, then antibody-dependent enhancement, or ADE, can occur. Occasionally, non-neutralizing antibodies and or complement components bind to the virus in the case of a viral infection and promote phagocytic cellular entry via the Fc gamma or complement receptors. Examples of this have been reported with dengue fever, which is a flavivirus, but have also been seen with feline coronavirus and feline coronavirus vaccines in cats with FIP. And inconsistently, in some animal models, the hamster and rhesus macaque of SARS coronavirus. Thus, ADE is a theoretical safety concern when developing a vaccine for SARS-CoV-2. Another point to consider when thinking about ADE is that enhanced disease may also be driven by the generation of a Th2 rather than a Th1 response to the vaccine antigen. So, what is the risk of ADE with vaccines targeting SARS-CoV-2? It appeared that the risk was low based on several factors. First, expectations were that COVID-19 vaccines would produce high neutralizing antibody titers and low levels of non-neutralizing antibodies. As more COVID-19 cases occurred, there has been no evidence so far of enhanced disease after SARS-CoV-2 infection and re-exposure or treatment with convalescent plasma. And coronavirus enhanced disease has so far only been demonstrated, as I mentioned before, with the feline coronavirus, which is an alpha coronavirus, while SARS-CoV-2 is a beta coronavirus. However, while the likelihood was low, it is still a potential risk, and monitoring for ADE was still warranted. So non-clinical efficacy models have been used to assess the potential for ADE, and in addition, clinical trials included mechanisms to identify signs of ADE. To date, there hasn't been any reported. In addition, vaccine clinical trials have shown a strong Th1 rather than a Th2 response. Now let's talk a bit about a vaccine modality that I've become very familiar with, the mRNA-based vaccine. How do these vaccines work? The mRNA encoding the antigen of choice, in this case, the spike glycoprotein locked into a prefusion conformation, is encapsulated into a lipid nanoparticle, or LNP. The lipid nanoparticle increases the half-life of the mRNA compared to injection of naked mRNA, and also works to increase cellular uptake. Once the LNP is taken up by the host cell, it travels through the endosomal pathways and eventually releases its contents, the RNA, into the cytosol. The mRNA is then translated and the protein antigen is expressed. Depending on the signals contained within the RNA and the expressed protein, the protein itself may be expressed on the cell surface, or the protein may be processed by the proteasome and peptide fragments presented via the major histocompatibility complexes, or MHC. This slide lists some of the safety considerations for an mRNA LNP vaccine. First, mRNA is non-infectious and non-integrating. It is degraded shortly after entering the cell, but can stimulate the innate immune system via toll-like receptors such as TLR7. mRNA can be modified to contain modified nucleosides, many of which are naturally occurring, such as pseudouridine, which work to dampen that innate immune response. Finally, where does the RNA go, and where is the protein expressed? This is driven by the LNP, which acts as the delivery device for the RNA. LNPs generally consist of structural lipids, such as cholesterol and phospholipids, an ionizable lipid, the complexes with the mRNA, and a pegylated lipid to allow for better stability and half-life. Of course, if any of the components of the LNP novel lipids are used, a safety and ADME assessment will likely need to be conducted. This figure shows the traditional approach to infectious vaccines development. And as you can see, that can take many years. Sequential investments are made along the way as more confidence is gained in the vaccine safety profile and efficacy. This approach was obviously not going to work to stem the worldwide spread of SARS-CoV-2 and its associated disease, COVID-19. In the U.S., the first step to changing the approach to vaccine development occurred with the declaration of a public health emergency related to COVID-19, which occurred in February of last year. This allows the option to authorize use of unapproved products. The criteria for granting an emergency use authorization includes the disease must be serious or life-threatening, the vaccine or treatment must show evidence of efficacy, the risk-benefit analysis must favor the use of the vaccine or treatment, and there should be no adequate, approved, or available alternatives. In addition to the EUA, a precedent for a rapid approach to vaccine development during a public health emergency was established by the WHO in 2018 when evaluating the potential Ebola vaccines. This approach emphasized the use of platform data, which could be used alone or with supporting interim in-life data to enter phase 1 clinical trials. While the WHO focused on viral vector-based vaccines, I've expanded the potential definition of a platform as follows. A platform is a modality in which components are comparable with the exception of the antigen itself. So that would mean the same adjuvant excipients, the same DNA viral vector or plasmid, or RNA platform, as well as the same delivery device. Later guidances issued by the FDA specifically on COVID-19 vaccines, which are included here on the slide, also supported the use of platform data as a way to accelerate development. In 2020, other global guidances and initiatives supported rapid COVID-19 vaccine development, including a workshop by the International Coalition of Medicine's regulatory authorities that supported use of platform data to allow for rapid entry into clinical trials, and from the WHO outlining a pathway for an emergency use listing for COVID-19 vaccines. As an example of how this approach was used, and in particular for our Pfizer-BioNTech COVID-19 vaccine, BNT162b2 or Comirnaty. This slide shows the non-clinical safety-related studies that were supplied to health authorities to support the IND or CTA, compared with the data that was supplied to support the EUA or MAA. For the IND and CTA, we leverage RNA platform, non-clinical and clinical data from candidate vaccines targeting other indications, in addition to interim dosing phase data from an ongoing repeat dose toxicity study with a number of COVID-19 vaccine candidates. This was supplemented with some protein expression biodistribution data. For the EUA and MAA, we provided final results from repeat dose toxicity studies that had been conducted, timing of the DART study results in the situation of the EUA, or in the case of the MAA, an audited draft report from that DART study. In addition, data from an NHP immunization and challenge study, which was our non-clinical assessment for enhanced disease, was provided, protein and LNP biodistribution data, as well as a small ADME package for the novel lipids that we used in our LNP. Although we were able to utilize an accelerated approach, I must emphasize that there were no compromises made to the safety evaluation of the vaccine. Non-clinical safety data, including platform data and immunogenicity data, was provided to health authorities prior to entry into the clinic. Many of the activities were performed in parallel on an accelerated path versus sequential, the more traditional path that I discussed earlier. And these included non-clinical, clinical, and manufacturing activities, as well as others. As well as we engaged in constant communication with regulatory authorities on the timing of delivery of non-clinical and clinical data to support filings as well as ongoing activities. This slide, showing a snapshot of the New York Times COVID-19 vaccine tracker, illustrates how far we've come, with approximately 10 vaccines already authorized or approved in various countries around the world, and several more close to it, giving us the tools to tackle the current COVID-19 pandemic. In summary, what I learned from my experience helping to develop a COVID-19 vaccine during the ongoing pandemic is that early and frequent interactions with health authorities was critical. Platform data can support clinical trial initiation, and this data may be supplemented with interim data for less well-characterized platforms. Any safety data from candidate vaccines or related vaccines in humans may be relevant. And finally, it also leads me to wonder if there are any other areas where we can use these lessons, such as for severely debilitating and life-threatening indications. My final slide acknowledges only a few of the other Pfizer and BioNTech colleagues that were involved in the non-clinical development of BNT162b2, as it's always a team effort. Thank you very much.